Hello everyone, my name is Kyle Hippe and I'm with Pacific Lutheran University, and today I will be introducing CaoLab Prolango II, which is a protein function annotation method from a sequence-based RNN. So first I'd like to give a bit of background on the problem. So the problem is the protein function prediction problem. That is, given an unseen protein sequence, we must determine its biological functions. The way we do this is through the gene ontology terms, and the gene ontology is represented through three separate trees which represent the biological processes, the molecular functions, and the cellular components. Currently we must do this through biological experiments, and there's a couple issues with this. The first being the time and resources required. This is a very resource and time intensive process, and it makes it infeasible to annotate large amounts of protein sequences quickly. Second, we must oftentimes do multiple experiments to determine the function. This is because that proteins don't act independently of their biological system, so we oftentimes have to run multiple experiments to get an accurate set of functions. So a computational method would help bridge the gap between the relatively small number of annotated sequences and the increasing amount of unannotated sequences. So the motivation for Prolango II is, how do we understand the protein function prediction problem as a translation problem? So to do this, we must establish an input language and an output language. So our input language would be a protein sequence. If we analyze a protein sequence, we can look for common words, which would be amino acid substrings of the protein sequence, and we can establish a dictionary of these protein languages, which we will refer to as the prolang. And then if we consider the meaning, or the second language, as the functions of the protein sequence, then we can establish our second language, which would be the Go language. So the protein function prediction problem has been solved in multiple different ways in the past, and I would like to go over a couple of these methods here now. So the first heuristic is BLAST, which is Basic Local Alignment Search Tool. So this is the baseline from which we compare new methods. It is exceptionally accurate when there are large sections of homologous regions in the target. This means that the sequence that we are trying to identify has lots of commonalities between targets or between protein sequences that are already in the database. And while this is great, it encounters issues when the target has low similarity in the database that we're searching. So in the example below, we have a target sequence which only has 4 other related sequences, which would make it a challenge to transfer annotations efficiently and accurately. Another heuristic is similarity based. So to do this, we search a database to find homologies from a set of functionally annotated proteins. This differs from the BLAST base, as it is typically looking for homologies in other types of areas. So on the example on the right, we have a phylogenetic similarity, which we are searching. And then from these similarities, we can infer the function based off of where it's grouped in this relationship. Next, we have the network-based method. And here we generate protein-to-protein or other interaction networks to graph related proteins. And so then we can infer uncharacterized protein functions through the unseen protein sequences associations in the network. And to do this, we are going off the guilty by association method. So anything that is closely related, or a new sequence that is closely related to a group of proteins which have certain functions, will automatically get assigned functions that relate to that group. And lastly, we have the information-based method, which uses other biological knowledge of the protein and components of the protein in order to infer the function. So in this example on the right, this group uses mRNA co-expressions, gene fusions, phylogenetic profiles, and other features to try and infer protein function based off of the input sequence. So before we get into the Prolango II method, I'd like to give a bit of background necessary to understand our method. So the first would be an introduction to recurrent neural networks. So recurrent neural networks are a form of artificial neural networks that utilize previous information to produce an output based on many related inputs. So in this picture below, we have a node A, which appears to feed inputs into itself. So if we unroll this, we can see that given an input X, we feed it into node A, which feeds it into node A again, but with a different input X1. So as we do this, you can see that we we can establish ordered relationships from which the RNN can analyze patterns. Also, I'd like to introduce neural machine translation. So this is an approach to machine translation that uses artificial neural networks to predict the likelihood of a sequence of words. Typically, this has been used to translate a sentence in one language into another language. And in this example below, we can see that words A, B, and C are used to evaluate the likelihood of the words W, X, Y, and Z. So the goal of this is to maximize the likelihood of the sentence W, X, Y, and Z based off of the input sequence. So in the end, we will have a model which can, with high probability, predict an output of words from a given input of words. So now I would like to move on to the Prolango II method. So for those of you who are familiar with Prolango, the original Prolango, some of the changes that have occurred since then are, first, we have moved to a character level RNN, which we use a encoder and decoder architecture to analyze character by character the input sequence. Secondly, we have moved to an ensemble method. So we evaluated the top 100 performing RNNs and combinatorially analyzed them to produce the maximum accuracy for prediction. In the end, this turned out to be 20 models used in the final ensemble, and we will go into that a bit further later. Next, we have changed the training data a bit. Instead of taking words that occur in the protein database, if they occur 1,000 or more times, we just take the top 2,000 words. We'll go into how we establish the protein language a bit later. Lastly, we handle sequences that do not contain any words that are in the top 2,000 of our fragment sequence database. We'll go into this a bit later as well, but the gist of it is we will get a probability-based estimation of the Go terms of any fragments that do not appear in the Go language, which we'll also establish a bit later. So some assumptions for Prolango II. First, we must assume that protein sequences are similar to human language. This allows us to use the neural machine translation and allows us to provide the functions based off of that. Second, we have to assume that the meaning of a protein sequence is its functions. We also have to assume that the patterns and conventions present in a sequence are consistent and can be used to identify the meaning or the function of the sequence. This allows us a platform to jump off of for when we establish our neural machine translation. So I would like to now talk about how we consider the protein sequence as a language. So if we consider a protein sequence as a sentence, then in the human language, any sentence consists of a series of words. In the context of a protein sequence, these words are substrings of amino acids. So when we construct our fragment sequence database, we use the most recent version of the UniProt database, and of those 536,000 sequences, we analyze any substring of amino acids ranging from length 3 to length 7, and then we produce a list of the most frequently occurring substrings of amino acids or words occurring in this database. From this, we establish the top 2,000 of them, and then that is our words that we consider our protein language. Now I would like to explain how we establish the gene ontology terms as a language. So as we described briefly earlier, gene ontology terms are 3 directed acyclic graphs representing the biological processes, molecular function, and cellular components. We perform a depth-first search on these trees, and we get a base 10 index for each term. From this, we convert each base 10 index into a 4-character letter string with base 26, which gives us a representation of Go terms in a string format. So if we take a look at these trees here, this is an example, concatenated example of each of these gene ontology trees. And once we perform a depth-first search, we would get a result that looks a little bit like this, where each Go term, instead of having a Go ID associated with them, and now has a character string associated with them. So this is a bit of a flowchart which describes how we train a RNN model for Go term prediction. So the first step is to determine the protein language, as we described earlier. So we have our UniProt database with 536,000 protein sequences, and we establish our protein language in the salmon-colored box up at the top. Next, we establish the Go term language by performing the depth-first search on the gene ontology trees, and we get a set of character strings represented in the blue box below it. Next, we have a encoder-decoder RNN method, which is described below here. So the encoder is an RNN that receives the protein sequence on a word-by-word basis. Each of these words are then encoded into a content vector. Then the decoder, which is also a recurrent neural network, receives the content vector and predicts one at a time the Go terms that are associated with the input sequence. From this, we can decode the Go words that are predicted into Go IDs, which then correlate to our functional predictions for each protein. Now, this only represents one RNN, and as we said earlier, our method is an ensemble method. So we have trained over 100 of these models and have ranked them combinatorially and have determined that the top 20 of these are responsible for are most accurate for predicting Go terms. So a bit of the architecture details. As we said earlier, this is a neural machine translator based on recurrent neural networks. Some of the parameters, we consider the max words of a protein sequence to be between 100 and 300, and will pad or select down to that range. We tested hidden nodes from 250 to 4,000, and also learning rates from 0.01 to 0.31. For sequences with no words, this is one of the new additions to Prolango II. We skip evaluating based on the RNN models, and we predict Go terms with the highest likelihood for the words that do not appear in the fragment sequence database. And as we've mentioned a couple times before, this is an ensemble method which arises from the top 100 performing RNNs. We achieve the best accuracy with 20 RNNs predicting the Go functions of a protein sequence. So for some results, we tested 253 sequences taken from the UniProt database. We evaluated them using the top N metric, which produces statistics for the top 1 through 20 predictions for each target. We evaluated the maximum similarity and the average similarity for each Go term in the predicted set to the ground truth Go terms. So here we have a maximum similarity. As you can see, the blue method, Panzer II, is a similarity-based method. It uses blast searching and performs very well. The green is a network-based solution, NetGo2. The red lines are sequence-based methods. The star is Prolango II. The pentagon is HMM Meta, which is a in-house method developed by CaoLabs. And with the pluses, we have DeepGo Plus. Down in the black, we have Superfamily 2, which is an information-based method. And as you can see, Prolango II performs very well in the maximum similarity for sequence-based methods, but can use a bit of work to catch up to the network-based and similarity-based methods. For the average similarity, we see a bit of a struggle with Prolango II. It does not perform quite as well as other sequence-based methods and barely outperforms the information-based method we have included, but we believe we can address this with a bit of future research. So some of our future research includes including different models in the ensemble. We have considered hidden Markov models as well as Bayesian neural networks. We also would like to tune different RNNs for molecular function, biological processes, and cellular components. Having a set of models that are trained only on each category could allow us to improve the average accuracy for each of these gene ontology trees. We would also like to include network-based interactions as possible. As we can see from the Panzer II results from before, network-based and similarity-based methods are very accurate when homology is high. So if we can include some of the information that is generated from those, we think that that would greatly improve our research. For some acknowledgement, we would like to thank Dr. Renzi Cao for his support. I'd like to thank Shola Benro for his time and resources. We would also like to thank Google Cloud Platform for their resources and PLU for their investment in us, as well as the National Science Foundation for their funding and support. Implementation details are available with the GitHub link, and the previous publication of Prolango is available at the link provided. If you have questions or comments, please contact myself, Kyle Hippe, at the link below, or Dr. Renzi Cao at the email below. Thank you.